So today in this video, we're going to be discussing denaturing proteins and the idea of chaperonins. And the learning objectives are, I want you to predict how the structures of two proteins may differ if they evolved in different conditions. I want you to be able to demonstrate how the changes in pH and the ionic conditions that a protein is dissolved in can denature proteins. And I want you to be able to explain why chaperonins are necessary for some proteins to properly fold. Proteins evolve under specific or in specific environments. So for example, we have Thermus aquaticus. They live at 72 degrees Celsius. We have E. coli. They live in our guts. And so therefore they evolved at 37 degrees Celsius. And both of them have this protein, a DNA polymerase. Okay, so they both inherited that protein via a common ancestor, but because they live under different environmental conditions, that protein is going to have evolved a slightly different structure to accommodate those different environmental conditions. So I have these two questions. How similar will these two proteins be, and how might they differ? Well, since the DNA polymerase from both Thermus aquaticus and E. coli came from the same common ancestor, they are going to be, those two proteins are homologous, and therefore they will be quite similar. They're both DNA polymerases, and they have to catalyze the production of DNA. But since they evolved in different conditions, they will be different. What might be a way in which they're different? Let's think about if we are at very high temperatures, all of those various weak interactions, hydrogen bonds, are going to be breaking much more easily. So can you think of an interaction that stabilizes the tertiary structure of a protein and the quaternary structure of proteins that is stronger than a simple weak interaction? The disulfide bridges. And sure enough, and remember, a disulfide bridge requires a sulfhydryl group, and those come from cysteines. And sure enough, if you take a look at proteins that have evolved in higher temperatures and compare them to the homologous proteins that evolved in lower temperatures, you will see a higher number of cysteines in the proteins that evolved at higher temperatures. So even though both DNA polymerases came from a common ancestor, because the two different organisms live in a different lifestyle, have a different temperature, those proteins will begin to evolve different structures. And what this means is that each protein has an optimal set of conditions. They evolved in a specific set of conditions. They will then have an optimal set of conditions in which they function best. The optimal temperature, an optimal pH, and optimal ionic conditions. What that means. So let's think about this. Well, what happens then if the environment strays from these optimal conditions? What levels of protein structure will be affected? Well, let's take a look here. Any structure of the protein that depends on weak interactions is going to be affected. So the quaternary structure, the tertiary structure, the secondary structure of proteins will all be affected when the environment strays from those optimal conditions. What won't be affected is the primary structure, because that primary structure doesn't depend upon weak interactions. It just depends on those peptide bonds connecting the adjacent amino acids together. And peptide bonds are covalent bonds, and just temperatures and pH and ionic conditions aren't going to really be enough. You'd have to really increase the temperature to start breaking down those covalent bonds. Now let's take a look. Temperature. Well, temperature is just kinetic energy, and the more kinetic energy you have, the more likely you're going to break those bonds, those weak interactions. But pH and ionic conditions, we can go into a little bit more detail. So let's take a look at what happens to a protein when you stray from its optimal pH. What happens to a protein when you stray from its optimal ionic conditions. In this video, I want to demonstrate why proteins have an optimal pH and why proteins will denature if you stray too far one way or the other from the optimal pH. So what we have here is a hypothetical protein. So this is a chain of amino acids, and two of the amino acids in this protein are lysine, and the amino group of lysine is shown here. That'll be the amino group on the R group. And aspartate. So this is the carboxyl group on the R group of aspartate. Now this amino group here in lysine has a pKa of 10.53, and this carboxyl group here has a pKa of 3.65. And let's assume that this is a protein within our cells that has an optimal pH of about physiological pH. So pH 7.3 or something like that. Okay. Now, when the pH is 7.3, let's take a look at how many of those lysines are going to be protonated. Let's see how many of those carboxyl groups, those aspartates, are going to be deprotonated. So let's focus on the lysine. The current pH of the cell is 7.3. And the pKa is 10.53. So that means the pH is at least 2 units less than the pKa. Okay, 2 units less than the pKa. And based on what we learn, when that's the case, when the pH is at least 2 units below the pKa, we know that 99.99% of molecules are going to be protonated. And because it's an amino group, it has a positive charge. Good. So we have it written up correctly here. Let's take a look at the carboxyl group. Now the pKa of that carboxyl group is 3.65. The pH is 7.3. So we have a situation here where the pH is at least 2 units above the pKa. And that means, again, you'll have to go and review the lecture, that means that 99.999% of the molecules will be deprotonated. In the case of a carboxyl group, that means it's going to carry a negative charge. Good. So what that means is at pH 7.3, most of the 99.9% of the amino groups are going to be positively charged. 99.9% of the carboxyl groups are going to be negatively charged, and we get this beautiful ionic interaction that forces the protein to fold in this way here. And it's this fold in the protein that is necessary for the proper structure and therefore proper function of this protein. Beautiful. Everything's working as it should as long as we are at the optimal pH. Now what we're going to do is we're going to take a look at what happens when we increase the pH and decrease the pH. So let's take a look at when we decrease the pH. So let's see here. If we take the pH and we decrease the pH by adding acid to the solution, what are we doing to the hydrogen ion concentration? We're increasing it, right? So we've got these protons. We're just increasing, okay? As we increase the concentration of protons, what's going to happen? Well, our NH3 plus is already protonated, so adding more protons, so decreasing the pH from 7.3 to 6.3 to 5.3, it's not going to affect the amino group. It's already protonated, cannot get any more protonated. But take a look at this. as we reduce that pH maybe to 6.3, and then maybe to 5.3, maybe to 3.3, and so on and so forth. We're adding more and more protons, and what's going to start happening is this carboxyl group is going to become protonated and lose its charge. In this state here, this carboxyl group, now that it's protonated, cannot interact ionically with this amino group here. We're going to lose this fold. And we even have a good idea of when this is really going to happen. It's going to happen at a situation really where the pH that we are bringing this down to is 2 units less than the pKa of aspartate. So once we get down to 1.65, nearly 99.9% of these carboxyl groups are going to be protonated because we have such a low pH, such a high concentration of hydrogen ions that even carboxyl groups that don't usually hold on to their protons very well, even they are going to be protonated. So at this super low pH, let's say pH 1.65, we know our amino group is going to be protonated. It was protonated a long time ago. But we now know, since we're 2 units here, that almost 100% of our carboxyl groups are going to be protonated as well. We no longer have that ionic interaction, and that protein is going to denature. Now, let's look at the other side. Let's look at what happens when we take that pH and we start to increase it. We start to look at that proton concentration, and it's decreasing. So now what we're doing is we're removing protons, we're removing protons, we're removing protons. We are, right, increasing the pH, and eventually, all right, we're going to get back to our optimal pH. Beautiful. 100%, almost 100% of the carboxyl groups have become deprotonated again. They've got a negative charge. Those amino groups are all positively charged because they're all protonated. Beautiful. But now, let's keep going. Let's keep increasing that pH. So now that we go to 8.3, and then we go to 9.3, well, as we do that, we're taking away protons, taking away protons, taking away protons. Well, that's not going to affect the carboxyl group at all. We've already taken away all of its protons right around pH 7. That was not a problem. But as we keep increasing, eventually what's going to happen is we're going to start to lose our protons from this amino group of lysine. And when we take away that last proton, we no longer have a charge. And now that we don't have a charge here on this amino group, we can no longer have this ionic interaction, and we're going to unfold that protein. It's going to lose its structure, and it's going to lose its function. On top of that, because we know the pKa of this amino group here, we know when this is going to get really bad. It's a point at which the pH is 2 units above the pKa. So when we bring that pH all the way down to, to 12.53, we will know almost 100% of these amino groups will also be deprotonated, and therefore we will not be able to have an ionic interaction with this negative charge on the carboxyl group, and the protein will unfold. It will denature. It will no longer function. So hopefully what you've got from this is that proteins have a optimal pH, and that's going to be the pH where all of the various weak interactions can occur. However, if you stray from that pH, if you go too high, if you go too low, you're going to start to mess up the ionization states of some of these important R groups. You're going to stop these weak interactions from occurring. The protein is not going to be able to fold properly, and it's going to lose its structure and therefore its function. Now, proteins also have an optimal ionic strength. So the solution in which they are dissolved has to have a specific ionic strength. And if you stray away from that too much by either not enough or too much, then there's the possibility of denaturing that protein. So here we have that same protein here, lysine and aspartate. There's our COO negative and our NH3 plus, and we have an ionic interaction there. Now, if we add a bunch of salt to the solution in which this protein is dissolved, let's say sodium chloride breaks down into these sodium ions and chloride ions, what will eventually occur are all of the positively charged ions will circle this. The chloride ions will interact with that positive charge, and what that does is it weakens the interaction between the carboxyl group and the amino group. And so this weaker and weaker interactions as a result of now the competing interactions with the sodium ions and the chloride ions are going to cause this to unfold and therefore denature. So let's make some definitions here. If that protein is in its functional state, we know it's in its native conformation, the structure that allows it to carry on its normal function. And if you do something to denature it, and if you stray away from its optimal conditions, it will unfold and become denatured. But as we know from the Anfinsen experiment, the information here in the primary structure that's intact, even though it's denatured, it's intact, is enough to allow that protein to renature into its native conformation, assuming you put the protein back into its optimal conditions. But here's the problem. Make sure you watch the unboil an egg video, because it shows you something that even though each individual protein has the information necessary to fold, and sometimes within a cell you've got so many proteins trying to fold, they tangle up with each other, and that doesn't allow them to fold properly. So sometimes cells need this artificial, isolated environment in which to properly fold so they don't tangle with others, and those are chaperonins. Chaperonins are large protein complexes, and they look kind of like a barrel. And in go the proteins that are not quite folded. That barrel offers an isolated environment, and out those proteins come in their native conformation. And so basically chaperonins are protein complexes that allow other proteins to fold in an isolated environment so they don't get tangled up with other things. All right, so make sure you watch this video again. Make sure you review it and are able to predict how the structure of two proteins may differ if they evolved in different conditions. Make sure that you can demonstrate how change in pH and ionic conditions can denature proteins, and be able to explain why chaperonins are necessary for some proteins to properly fold.